I've been a botanist for 48 years. I know there are certain people in this room who've been botanists for even longer. But if you've been a botanist as long as I have, you will have grown up with the Scrophulariaceae as this assemblage of plants here, ably illustrated by Keble Martin. And then, in the last couple of decades, this happened. They were shattered into different groups, and then to add insult to injury, the plantains were stuck onto them. So the title of my talk is Divorcing the Scrophs, How Molecular Data Impacts Classification. So I'm going to, in about 20 minutes, I'm going to try and go through how molecular data works in a very simple way, how and why it can conflict with existing taxonomy, and the effects it's had on selected families of Scrophs and Lilies. The aim being to leave 20 minutes to 10 minutes, sorry, to have what could be a very lively discussion. Okay, so molecular data. I mean, this is actually really, it's both complicated and simple. So the actual analysis can be very complicated. You can be looking at thousands and thousands of characters across countless individuals. So I've simplified this almost, but not quite, to destruction. So I'm just going to ask you to imagine a hypothetical group of 8 species. And in this case, we are looking at the DNA. And again, for simplicity, I'm asking you to imagine, first of all, they all have just this little stretch, and it's all the A DNA base, which, of course, wouldn't tell us anything about the relationships. But if we have a situation where 2 of them have a T on their second base and these 3 have a G on their fourth base, well, that tells us something, because the analysis tends to assume the fewest possible changes to give you the observed spread of DNA. So it would assume that these 2 at the top, a single mutation at position 2 to give you a T, here a single mutation at position 4 to give you a G. So these 3 appear to be more closely related to one another than they are to any of the others. And if we add in some more changes like that, then you can see how the more characters you have, the more information the tree contains. And in this way, we can use molecular data to build up a picture of how they are related. But this is slightly misleading, because we don't at the moment have any idea of what the common ancestor looks like. We've sort of assumed that it's all A's, like so, but we don't know that. So, for example, the common ancestor could look like that, with a C at position 6, in which case it would fit there onto the tree. Or it could have a T at that position, in which case it would fit there. And these have different implications for exactly what is closest to what. It could equally have a T there as well, in which case it would fit here. Oh, it's missed slightly. It's something has changed, a small thing has changed in the transference, but never mind. I think you can see the point. Okay, so which is it? Which is correct? Well, we can't go back in time and sample the common ancestor, but there's something we can do that's almost as good. So if we shrink that down to the corner there, we can effectively move backwards to the ancestor by moving sideways to the a closely related group like this. And because basically you're doing the same thing, and by including these in the analysis, you can actually get a pretty strong indication of what the ancestor was doing. If you go back further, you may be able to strengthen it further by looking at the next most closely related group as well. So these are called outgroups outside of the main study group, and it allows you to put a direction on all of the changes that have happened in the DNA. And so in this way, we are able to objectively, and this is an advantage of molecular data, it is totally objective. It's done by computers. They do not have feelings. They do not have preferences in the way that we do. And so they will give you an objective account of what is related to what. But we have grown up with taxonomy, and sometimes they've come into conflict. So this is a hypothetical group of 11 species. I'm going to use these to explain how and why molecular data conflicts with existing taxonomy. So here they are, and if you put these in front of a taxonomist, well, taxonomists would probably divide them up, perhaps a bit like this, into sort of 4 groups of similar species. And in particular, these 3 over here, you can see they've got different coloured flowers and they are climbers, so they're more different than the others. So let's imagine a taxonomist might call this Bryonoides and call these Bioloides, and perhaps have 3 little subgroups, maybe subgenera, like so. And that would be the taxonomy. And all that might remain stable from the time of Linnaeus through to about the 1990s or the 2000s, when molecular data came along and changed it. So sometimes, even before molecular data, people would sometimes try to use sort of morphological characters to build a phylogeny in the same way as molecular data. So we might assume that the relationships look like this. Then let's imagine molecular data. It might come and show you something totally different, like this. And you go look at this and you go, what? What? Why are these closer than those? But actually, there's a perfectly sound reason why this would be the case. So you get things like this happening because the defining characters for all of these Bioloides may be the ancestral characters for the group. And we're going to see this in these 2 families where all these changes have occurred. So if the common ancestor looks like this, well, we can map on the different characters and how they've changed and where they've changed. And actually, I've been involved in work on the entire nettle family, Urticaceae, where we first built a giant phylogeny for the whole family, and then we were able to map morphological characters onto it, determine which ones changed only once and which ones changed multiple times, and therefore understand which morphological characters are better for classification. But doing it here, what you notice is there's lots and lots of changes there. If you know your Darwin, you may have heard of punctuated equilibrium as a way he described evolution. And this would be a punctuation. So you can see sometimes you get these single lineages where there's a big, rapid change. And that's how you may get something that morphologically looks like a different genus deriving within something that morphologically looks like a first genus. So everything with these purple lines would have looked broadly like this. And then you get this sudden outbreak of massive, lots and lots of changes, giving you something that looks totally different. And the key thing is, for Bryonoides, the traits are derived along that arrow. The shared traits for Bioloides are ancestral to the whole group. And this is why DNA can conflict with morphology. What do we do about it? Well, first of all, does this matter? Is it at all important that that is more closely related to that? I'll give you 2 reasons why it might be. So first of all, if we start to get a new emerging disease and it's attacking these ones, and you think, well, you want to think, well, which is it going to attack next? The genetic data tells us that these are more vulnerable than those, which the morphological data doesn't, because they're genetically more similar, even if they don't look like it. Likewise, if somebody were to discover a potential new drug for human health in one of these clade 2, but maybe there's side effects, you want to find something similar but not quite the same. Again, the best place to look would be there, not there, as morphology would indicate. So these things do actually matter. What do you do about it? What's the taxonomic response? Well, firstly, you don't want to do this, okay? You don't want to do this because it just makes it impossible to separate these 2 genera in any meaningful way. So you don't just blindly follow the molecular data, you've got to use a bit of intelligence. So you could simply synonymize them all into one monophyletic genus and solve the problem that way, call them all Bioloides. You could erect a new genus for this little clade here, and that way only 3 of them get their names changed. Perhaps this is the most elegant solution of the 3. And you can see then that this new genus, whatever you call it, is understood to be more closely related to Bioloides, although it looks more like Bioloides. Finally, you could maintain the status quo, but there is that fact that it is misleading. Anyone who hasn't looked at the phylogeny won't understand the relationships, which could matter if the implications become important. We can discuss this at the end. But let's now look at what it has done to 2 of our beloved families that we grew up with. So petaloid monocots that occur in the British Isles, there's basically these 4 families that are in Keble Martin that we are all familiar with, my age group and above. And then molecular data came along. And here's what happened. Well, the herb Paris, which has kind of been in and out of the Liliaceae even before molecular data, that goes into Melanthiaceae, which is not that far from Liliaceae. And you can see the colchicum, the so-called sort of Menosaccharum, that's also a bit distant as well. But what about the bluebells? And other things as well, like the grape hyacinths are in that group as well. Well, bluebells actually are much more closely related to daffodils. Our garlics, alliums are basically with the daffodils as well. But look how far away they are from the lily family. To get from the lily family to the bluebells, you've got to go all the way back to the common ancestor, way down at the bottom here, and come back up again. So, first of all, this is why we've got Colchicaceae now erected to contain the Menosaccharum. And what else have we got? Well, this dotted line is how far you've got to go to get from the bluebells, which are now placed in Asparagaceae with the asparagus and various other things. The Camellids, this is grasses, pineapples, palm trees. These are now actually closer to bluebells than either are to lilies, remarkably. And it's because probably the common ancestor here may well have been a pretty bulb looking somewhere like a bluebell or a lily. So we've got Xanthoriaceae now to contain the red hot poker. And in fact, the lilies are now in a different order to the bluebells. So really, I mean, in this particular case, the molecular data shows that they are so different from one another that really the Liliaceae, as we used to understand it, it really can't be maintained. And if you think about it, it's always been a bit of a dustbin family. It's basically anything that's pretty and a bit bulb-like that isn't a daffodil, isn't an iris, and isn't an orchid. So I think we have to let the Liliaceae go. Okay. So there we have it. The Scrophs, well, the story in some ways is similar, but there are differences as well. So let us get into that. Same as before. Here we have the Scrophulariaceae, as I grew up with it, and other things with bilateral flowers that are familiar in the British flora, and 5 different families there. Let's see how they are related. Well, there is the phylogeny showing us that there are 4 different evolutionary lines going into the Scrophulariaceae. The other families mostly hold up pretty well. And this dotted line, again, shows us how far we have to go back from things like the red Bartzia there to get to a common ancestor with the Verbascum, and even further to get to a common ancestor for the Scrophulariaceae. So really, the Scrophs, the sort of characters that united the Scrophs, are effectively characters that are ancestral to this entire clade of things. So the Organocacea were defined by being oviparasites, Laniaceae, the labiates have their own defining characters, Lentipulariaceae obviously eating insects. So we've got these well-defined small families that Scrophs effectively defined by things, characters, ancestral to this entire clade. So this is why the Scrophs have been broken up into these 4 different evolutionary lines. And we have given these each new names or old names, depending if they've got one or not. But look at how close the red Bartzia is to Organocacea. So that's been moved into the Organocacea. So let's just have a quick look at this because it's interesting and informative from an evolutionary perspective. So this is the Organocacea as it has now been recognized. Everything in it is either a semi-parasite or a full parasite. So once again, oh, God. Okay. Once again, this is the original situation 6 years ago, Scrophulariaceae, Organocacea. Now, you may recall maybe 20, 30 years ago that the Lathraea's toothwort were actually moved into Scrophulariaceae. It was worked out that they are much closely related to these things. In particular, I think they look very like red Bartzia. If you compare the flower morphology, they do look very similar to me. And then later on, all of these were squashed back. Back you go, come on. Oh, maybe it's a kid that leaves home and then can't afford a house and goes back to live with his parents. Anyway, what actually happens from an evolutionary perspective is this. You have a heterotrophic ancestor, sort of general scruffy thing. It evolves to become a semi-parasite. Now, think about it. Evolving to be a semi-parasite is difficult to do. You've got to evolve an entire system for stealing photosynthate from another organism. It's difficult. But to go from being a semi-parasite to being a full parasite, all you've got to do is lose the ability to photosynthesize and not die. And everybody here at some point in their life has lost something, and everybody here so far has not died. So it's very, very easy to do. And so we have one origin of being a semi-parasite and multiple origins of being a full parasite from an ancestor like that. So Lathraea, separately from Organocacea, became a full parasite. So it all actually makes sense. And we have this reduced Scrophulariaceae with only about 4 or 5 genera. They have acquired Budleia as a sort of booby prize for all of what they lost. But that's not quite the end, because we have, what do we call this clade here, with the foxgloves, snapdragons, speedwells and stuff. Well, thing is, it's also, according to molecular data, Calitriche belongs here, mare's tail belongs here, and so do the Plantaginaceae. And of these things, well, let's just connect them all up, first of all, because molecular data tells us they are all one family. There you go. Let's colour them all the same. And what's the oldest published name for this family? Well, it is Plantaginaceae. And therefore, if we're going to go with the molecular data, unless we split it up into lots and lots of little families, we have to call it all Plantaginaceae. And oh dear, it's missed. Yeah, I think it's an issue. I think it's an issue of screen size, screen shape. Anyway, so it's meant to be sitting over the Plantaginaceae, but somebody didn't like that, and Clive Stace decided to rebel. Look there, he's rebelling, and he said no. I refuse to call it Plantaginaceae. This is too confusing for the beginner botanists out there, and indeed the experts out there. So I'm going to keep these families separate. And that is basically where we come to. So he's invented this name, Veronicaceae, to go for these and for what would be Plantaginaceae, but with these things excluded because they look so very different. Therefore, we are left with this compromise, at least if you follow Stace, where Plantagenet is not allowed to join the party because Stace doesn't like it. And I think this would be a good time to open up the floor to questions.
Thank you, Richard. Microphone. Thank you, Richard, for a characteristically entertaining talk. We have 5 minutes, I'm afraid, not the 10 you were hoping for.
Oh, well.
For questions. Who's going to go first? Down here.
Thank you, that's fantastic.
Right.
Given the biogenetic trees that you showed, the family tree,
Yeah.
what sense does it make for us to think in terms of gender and families and things like that? Because clearly these ancestral lines all go back different timing to the original.
Oh, yeah. Well, this is a big question because, I mean, you're probably all familiar with the kingdom, phylum, class, order, family, genus, species. Well, species is only one of those that really has any biological meaning. The others are all just human constructs. The problem is the angiosperm phylogeny is massive. And I think the class kind of wanders around. It doesn't really fit because you've got this massive tree of angiosperms and then these rather small clades of gymnosperms next to them. So that the whole hierarchical thing somewhat falls apart. Genus and family are basically, in my opinion, they are names that we use in order to put things into manageable groups. I mean, you may notice that binomial names are not limited to scientific names. We all have them. Cars have them, Ford Anglia, Ford Prefect and stuff like that. It's surprising. And of course, in China, they've evolved the same system independently as well for names. So yeah, genera and families, they exist basically to help us. And so we should treat them in that way, that we should use those names in a way that helps people as a compromise between morphology and practicality on one hand and biological reality on the other. And people may have different ideas where that line should be drawn, but the user, I think, is what you have to think about.
Another question? Yeah.
So,
Rich, could you just wait for the microphone so that we can record it? Thank you.
So, that being the case, it sounds like it's time for a new system. Right.
It's very easy to say we should have a new system. It's always a lot harder to say what that system should be. Plus, if people are going to complain about the changing the family name for a few dozen members of the Scrophulariaceae, you'd probably get a bigger amount of complaining if you were going to change the whole system. I mean, I think generally the naming system we have works. I mean, the fact is, if you look at everything, all the other families in Keble Martin, most of them were barely affected by the molecular revolution. There were bits here and there happening, but generally, I mean, I picked out the two where the effect was the biggest. Most families that we grew up with have weathered the taxonomic storm of molecular data. Also, it's more good news is that I don't think we'll see this kind of change ever again. I think molecular data has told us what's related to what. So we're not going to, once we get used to the changes that have happened now, we don't think we're going to get more to the same level. So that's only a partial answer. But if anyone comes up with a better system, I'll be interested to hear it.
Well, I mean, there is the phylum code, which is a proposal to rename everything in the living world entirely on phylogenetic basis. What do you think of that?
I don't think it'd be popular. Although
I have a feeling that that's already been decided.
I think you can get away with it as soon as you go away from visible plants and animals and maybe fungi. If you go down into microorganisms, only scientists really work with those. So I think it's fine. It's just you don't want to go annoying all your user base.
Great talk, Richard.
Most of what you've described has been based on barcoding DNA analysis. Now, more and more species have been fully decoded and got fully sequenced. Is that going to have an
I'm not at higher levels. I think there's enough DNA. I mean, generally changes are only made when the statistical support is higher. And if you go down family relationships, generally with the fragments of DNA that were used, there was enough support to say, yeah, this is definitely how things are. Where this sort of whole genome stuff is going to be very, very important is when you have critical groups and rapidly radiated groups where, so if you've got, say, one lineage and then a very short period of time splits into lots and lots, that Darwin's finches probably did on Galapagos, that then you, because it's sort of, if there's a very small bit of branch between one junction and another, then there'll be very few mutations along that branch. And to pick up that information, you probably need to sequence the whole genome. So generally, the whole genome is going to be very useful for critical groups, rapidly radiating groups, groups that hybridize like crazy, like Dactylorhiza and eyebrights and stuff like that. It's going to be very important for telling us stuff, but I shouldn't be too worried about it completely changing relationships at this level.
Building on RBG, you could adjust this situation by a simple change of nomenclatural rules. Because if you put the plantains into the Scrophs and call them Scrophs, far fewer people would complain.
Well, you
just don't like the idea of foxgloves becoming plantains. So just the system of conservation.
You could
conserve the Scrophs against plantains.
Well, you could call them Scrophs because the Scrophs are the mullions and the Scrophid area, but you could sort of, they could decide to call them all Veronicacea. They could do that. I mean, I don't mind the name Plantagenaceae. It makes me think of the Plantagenet kings and the days when Britain actually mattered in the world. But so yeah, I mean, it's an interesting one whether the actual chosen name matters. To me, I'm quite happy with Plantagenaceae for the whole thing, but it's the sort of thing that could be done democratically. And I'd agree with. I mean, there are cases certainly when I think names should be chosen, kept according to what's familiar, not the golden what was published first rule. And I think there should be more flexibility on that. On that point, I agree.
Richard, this is done at Botanical Congress, isn't it?
I think so.
5 years. And is it done by voting? Is it actually democratic among botanists?
I don't. Has anyone here ever been in one of those meetings?
And what?
It ends the complication.
Okay. So it's like you don't want to know how sausages are made. That sort of thing.
People would take a very strong line about changing the rules in that way. Yeah.
Yes.
It
Yes.
would be popular.
Yeah. There's a horrible scandal about acacia, isn't there? We won't go to that. Right. One more question.
How much do you think? There are lots of taxonomic characters. Some of them are going to be of more standing phylogenetically than others are. How much do you think the phylogeny can help us pick out those characters we should really concentrate on to distinguish plants from other things which are really not very important?
A lot, but it's not going to be the same in different groups. So what may be highly labile within one family could be very non-labile within another. So I mean, this paper I worked on Urticaceae, we could find things that were changing back opposite to alternate leaves and stuff like that. The sting only evolved once, but it was lost twice. You really have to look within a group to which, once somebody's done the phylogeny, it's not that difficult if you're good at your job. You just map the characters onto all of the species that are in the phylogeny, and then you do the same kind of parsimony process going down, and you work out which ones have changed once, which ones have changed multiple times, and that tells you which characters are more useful in that group. But you can't assume it's going to be the same in another group.
Okay, thank you very much. We'll